Right now, centriole in electron micrograph. Here you can see a centriole. Look at these two lines. I mean, it's not 100% clear because they are very small centriole and they don't have a very strong structure to see in the electron micrograph. But we can see a little bit like a wheel-like a structure. I think this one, right? This one here, just like a wheel-shaped structure, and a little bit here, it's like a tube-like a structure. So why we have two things? Why we have two different shapes? So both are centriole. That means I can say this is a centriole, and the other one is also centriole. This is centriole, and this is also centriole. Then I can say two centrioles. In a cell, we have two centrioles. So let's look, maybe not the electron micrograph, because the electron micrograph, it's important to remember the picture of electron micrograph. But if you want to make a little bit more clear picture of the drawings, maybe help like this. Now you can see again two centrioles. Together we call a centrisome. Two centrioles make centrisome. And this two centriole arrangement is also very special. One is sitting like this, and 90-degree angle, the other one is located. It's like a T shape. The T letter shape, the orientation, and they're together. And two centrioles make the centrisome. So in a cell, we can see two centrioles. That's what very clear in the electron micrograph picture. This one, one is sitting like this, other one is standing like this. So that's why we have these special shapes, and two centrioles make this orientation. Right now, coming back to the structure, again, microtubules coming. Again, microtubules coming. So now you know this yellow color ball, that is a protein make the microtubules. So tubules help us to remember the protein tubulin. You can see here three tubulins. Three tubulins. And now the three tubulins line up. I cannot draw it here. They line up to make a one long line. And then a second one is also joining. Third one is also joining. So we can see in 2D picture, three tubulin molecules. But if we get a 3D picture, one behind another one, behind another one like that, tubulin molecule line up behind this, each three of these tubulin molecules. So it makes a little bit longer line. Right. So example, if you look at this picture, here you can see the length. This length is due to the lineup of one tubulin with each other. Here now we can see 2D picture, only three, but behind it there are more tubulin molecule line up. Right? Then another one is coming very closer, like this. Yeah, like this. Now you see three long tubes or pipes, let's say, 3 tubes. Join in a circle, and we can see 9 triplets. So we call the 3 is called triplet, 9 triplets. So 9 triplets line up to make this centriole. And there is a more advanced structure of centriole we can learn, because you can see blue color, another one blue color in the middle, the wheel-like things. But we are not going to learn the small molecules which actually support these centrioles. It has a little bit more information, like with more proteins joined together to make different structures. But we don't going that far. We just say it is microtubules, triplet of microtubules, and then 9 times it's going around it. Yes, function is, you can see a centrosome here. One end of the cell, there are 2 centrioles, and the other end there are 2 centrioles. Then in this picture there are 4 centrioles. 4 centrioles or 2 centrosome. 4 centrioles or 2 centrosome, because 2 centrioles make a centrosome. And then they make these microtubules. So they are the organizing center for microtubules, and they form microtubules. And you can also see chromosomes attached to the microtubules. So one centrosome to another centrosome, there is a lines. These lines are microtubules. So when we learn in cell division, microtubules in this kind of organization, we're going to use another name called spindle. We call spindle microtubules, but we use the word spindle. So spindle try to organize the chromosome, and then after that the chromosome move to one side and to the other side. The centriole is the major microtubule organizing center during cell division. So they are important during cell division. They are very important in cell division. Okay, slightly related structure to centriole. I think I should include a very important thing here under centriole. Maybe I think I missed that one, right? So one important thing of centriole is we don't see centriole in all organisms. We don't see centriole in plants and fungus. This is a very important point to remember. We don't see, there's no centrioles in plant and fungus. That's a very important point. No centrioles in plants and fungus. Generally, other organisms, eukaryotes, they have centrioles. Right now, again, microtubules coming up. So microtubules, we have learned flagella and the cilia. Because cells can move using cilia or flagella. Bacteria, they have cilia. Bacteria, they have flagella, and sperm cell have flagella. So flagella is made out of microtubules. Centriole is made out of microtubules. Here again, cilia and flagella. Okay, that's a centriole. Right, we see a flagella. The flagella is attached to the cell membrane. The part which attached to the cell membrane is called a basal body. Because here you can see at this end, the bottom part of this screen, a basal body. That is the attachment to the cell membrane. From that attachment, the organization of microtubules make the filament. So we can say the flagella has two parts, basal body and the filaments. Other structures, like you see here in this picture, not necessary. Junctions on hook, stators, like this kind of thing, they are not necessary. So basal body is the attachment to the cell membrane and rest is the filament. This makes the cilia. Now, if you look the organization of microtubules, there is a different. There is different. What is the difference? Now, if you look this microtubules, the round-shaped structure, there are 9 around it. A pair of 9 microtubules. So they are not in. There are 2 now. Here it's a 2. And 9. Again, they make a 9 around this to make the ring. And you can find also in the middle. There's another 2 in the middle. This is usually written as 9 plus 2. 9 plus 2. So where we can find 9 plus 2? So we can find 9 plus 2 in the filament region. Filament region, we can find 9 plus 2. What is 9 plus 2? It's a pair of microtubules. Pair of microtubules make a ring. There are 9 around it. There are 9 around it, and it makes this ring. And in the middle, again 2. That's a 9 plus 2. We can also find this structure at the basal body. And here you can see again 9 plus 2. Make this microtubule 9 plus 2 the cilia or flagella. But centriole, it has triplet, not double, not pairs, it's triplets. Triplet 9 and nothing in the middle. There's nothing in the middle, triplet, 9 triplet. So here 9 duplex or pairs. That's the difference between centriole and cilia or flagella. Cilia and flagella, structurally same. The difference is cilia is shorter, flagella is longer. But microtubule organization is same. Right? So that should be the difference between cilia and flagella, right? And that would be different between centriole and cilia or flagella. Here the centriole, it has 3 microtubules in 9 times around its axis. And the other one, it's just 2. 2, 9 times around its axis, and middle also another 2, 9 plus 2. And what is the difference between basal body and filament? Here, basal body and the filament. The basal body is again the organization of microtubules. Filament is also again organization of microtubules. Then why we call filaments? Why we call basal body? Okay, basal body attached to the cell membrane. Filament is the free, the part of the flagella it can move. Is there any difference between structurally basal body and filament? Yes. Filament has 9+2 structure. Basal body has 9+0. There's no in the middle. We couldn't find 2 in the middle in the basal body. Here you see this is 2. This 2 is the in the middle. They are unfused. Unfused, 9+2, they are not fused, but others are fused. 9 fused, others unfused. And in the basal body, this 2 is missing. In the central 2 microfilaments missing in the basal body. So these are the very important difference between cilia, flagella, and centriole. They look more or less similar, but there are small differences structurally. Okay. Next organelle is vacuoles. Right? Does anyone know what is different between vesicle and vacuole? Because previously we learned vesicles. Because we have seen RER, they make vesicles. Vesicles is going to the Golgi. Golgi make vesicles. Again, Golgi transport the vesicles to the out of the cells or maybe into the cells. So we learned what are vesicles previously. Now we are learning vacuoles here. Okay, the difference is vesicles are smaller. Vesicles are smaller. Vacuoles are large structure. Vesicles contain vesicles contain protein and
So you are muted.
okay, thank you. Thank you. Mic is muted, yes. Yeah, I think what Chitsara said, vacuoles, they can't move from organelle to another. Actually, certain things what we are learning, there are little confusing areas. There are no clear definitions for certain things. So we don't want to compare vacuoles and vesicles, but if we want to know what is the difference, then because vesicles are membrane bound particles which has something inside, and they mostly used for the transportation. Yes, Chitsara is correct. Then they are more the functionally toward transportation, but the vacuoles have different functions. Vacuole function is different. They are not mainly for transportation. They are more for the storage, right? They are more for storage, but depend on again type of vacuoles. For example, when we take one vacuole in plant, That's very important, because plant vacuoles are very important, because they have a very clear, large vacuole, right? Okay, here, first vacuole I'm going to show. What are the type of vacuoles then? So vesicles, we just say secretory vesicles. They just packed with something and moving from one organelle, maybe to the other organelle, to the cells, cytoplasm, or out of the cells. They do transportation of material. They're smaller. But vacuoles are quite different. Their function is maybe different from one place to another place. Let's say this one. Okay. So we have to learn in the future this paramecium like protoctist, like it's a single cell, and they have two nuclei, right? They have two. One is called big nucleus, that's called macronucleus. Other one is called micronucleus. You see, that's a diversity of organism. So far we have learned mostly there is a one nucleus, but sometimes some organisms, they have two nuclei. One is bigger one is small one. Okay, we have to anyway learn the structure later on, but here I want to show you this contractile vacuole. That's called here contractile vacuole. Here one and here another one, right? This another one here. Is this one? It's a contractile vacuole. Now these organisms are single cell, and they represent different kingdom. Right? So all the organisms, there are 5 kingdoms. One is prokaryotes, the bacteria, so one kingdom, and then the protoctist, plant, fungi and animals. Protoctist, fungi, plant and animal. There's a noise coming. Just give me a second to close the window. Okay, live in a different place, different city. Right, this is a contractile vacuole. Now, why contractile vacuole is important? It's like this. If you look plant cell has cell wall. But this organism, it doesn't have a cell wall. And they live in water. The habitat is water. It's an aquatic organism. You will learn in plant unit 4 plants, a very special function of water. What is the very special function of water? Water molecules is moving from high water concentration to low water concentration. It's the nature of water. And we further clear this thing, high water potential to low water potential. The nature of water is moving from high to low. What is high water potential? High water potential means pure water. Water get more and more pure. That means less things dissolve in it, they get more pure. So they can move in. But if you look inside the cell, 70% of water, and there are many things dissolved in the water, because cell needs so many nutrients, so many ions, so many other molecules. They are dissolved in water. That means inside the cell, water potential is less, because solutes are more in the cell. But in an aquatic normal space, outside is less molecules. Then what is coming from outside to inside? So if water comes without any restriction, the cell can burst. Water coming into the cell, and cell can burst because it doesn't have a cell wall. If we consider plant cell, they have a cell wall. They can control the amount of water coming to the cell. For example, you take a polythene bag, thin polythene bag, and then you can fill with this water. You can fill with the pressurized water, it can break. If you a little bit pressurize, the polythene bag breaks. Now let's say you place the polythene bag in a container, a plastic container, and then you fill the water. But the plastic container supports the polythene bag. So it expands, and then the plastic wall hold, it doesn't go more beyond that point. You can't fill more than that. So plant, they have this function, but this organism, they don't have. Instead, they have this pump contractile vacuole. When the water comes, it can move excess water out of the cell. So they are continuously working, excess water pump back. That's a contractile vacuole. Anyway, we are learning this organism again in detail. So we will see again contractile vacuole there. Right, this is plant cell. Plant cell has cell wall. Okay, I think the vacuole in the plant, that's very important part of the plant cell. Because if we look at the plant cell, the size of the vacuole is quite larger. So we can say it's the largest organelle in the plant. So we try to say generally what is the largest organelle in a cell? It's a nucleus. We have seen the nucleus is very close to a 40%, 50% of the cells. But this time is different. Vacuole is the largest organelle in plant. Because of that, the plant vacuole, very important thing to learn. And the membrane of this vacuole, it's a single membrane. And the single membrane has another name for that. Because of this importance, the membrane get a name called tonoplast. Tonoplast is the vacuole membrane. That's a vacuole membrane, right? Tonoplast. Now, why vacuole is important for the plant? When we learning a plant, we have to again and again learn vacuole. Its role in doing so much of function. Number one, we can consider storage function, because large amount of water store in the cell inside this vacuole. When more water is stored, it gives a strength to the cell. So you can take an empty bag and you can blow air, and it will close it up. You can take again polythene bag, you can fill water and you can close it up. So you can see when it filled with water, it's more heavier and it's more dense and it's also more stronger. So the cell is very stronger and the pressure inside cell is quite again going up. In plant, we call this turgidity. The special word is used called turgid. I mean, the strong, right? We learn strong, strength. When it comes to plant, turgid. We use that word turgid. That's how it's biology, some of the terminology is coming. Mostly in plant, we don't use vacuole give the strength. We call it turgid. Turgid is the pressure given by the water. So it store water. On the other hand, it gives the turgidity to the cell and to the whole plant then. So plant can stay upright because it's attached to the soil and it's going up. Chitsara. Yes, the word herbaceous. So we are again going to learn plant. Let's say take a tomato plant, take a chili plant, and take a mango. So if you take these two, the difference is if you take a tomato-like things, they are soft. The stem is soft. If you take a mango-like plant, the stem is hard. You will learn this different. Some plants, they have a primary first type of growth, primary growth. Some plants, they have a primary plus secondary growth. The trees, they have a secondary growth. They are very stronger because the substances they have. But herbaceous plant, they have primary growth and they are very softer. The softness is mainly given by the turgidity of the water. And also you will learn the vacuoles store the nutrients, vacuoles store the pigments, vacuoles store ions, vacuoles store water. So these are the things stored inside the vacuole, and it gives the turgidity to the cell, turgidity to the plant, and it also regulate amount of water inside the plant. You will see very specifically the role of vacuole in plants when we are learning structure of plants. Example, the opening of the leaf, the stomata, closing and opening. Vacuole play a big role. So here, when we are learning unit 2, again it's repeating specifically these functions. Certain things, you don't worry too much, but some structures, only that place we're going to learn. But many things, we're going to learn again and again in some units. These plants, we're going to learn vacuole in greater detail and specific functions. So they are not that much important in unit 2, because specifically we're going to learn vacuole functions. Again, nucleus. Again and again, we're going to learn nucleus. So at many places. But microtubules, cytoskeleton-like thing, you have to really learn at this place. They're not much coming again and again, right? Right, we have again, you see the same thing is coming now. So pseudopodia, now you can, now actually the more you remember, more you understand when you learn different ways, different areas. For example, if now I think you can still remember if I ask, what is the filament responsible in making the pseudopodium? Pseudopodium is the singular, pseudopodia is the plural. Now, if I ask again, what filaments make the pseudopodium? See now, things are coming when we repeat it differently at different places, learning again and again, you get very confident, right? So we learn under microfilaments actin, pseudopodia making is important. So they play a role in making pseudopodia. So I think even last class and this class, we heard pseudopodia at least 10-20 times. Now you remember it. And we learned phagocytosis many times. So you remember it well. But we are learning for different purpose. Here, they take these particles, and then after that, these particles, they make this kind of the cell membrane coated a vacuole. We call food vacuole. So contractile vacuole, plant, the permanent large vacuole, and the food vacuole. There are 3 types of vacuoles. But we learn this phagocytosis. We are going to learn again this phagocytosis. But here we learn the structure made by this kind of a thing is called food vacuole. It's also considered as a kind of vacuoles because it's a large membrane bound, and now it's called food vacuole. Some animals we can find these food vacuoles, right? Okay. Right now, some of the things are extracellular. What does it mean? It is not within the cytoplasm. It is out of cytoplasm. Then, what about this cilia and flagella? They are out of the cell. But you have to remember, cilia and flagella, they have a cell membrane, right? So they have a cell membrane and they have a cytoplasm. Inside the cell membrane and cytoplasm, packed with microtubules. It's not out of the cell, it's inside the cell. So when we learn this cilia and flagella, it seems to be like outside, but it's a inside structure because cell membrane extended and packed with the microtubules. But you will see a small difference when we learn in prokaryotes and eukaryotes. That's why I kept to compare prokaryotes and eukaryotes at last.